Okay. So we are now learning this rat experiment at a different context in this scientific article. So next thing of this rat experiment, differences in the rat mitochondrial proteome were also observed when comparing the kidney, liver, heart, skeletal muscle, and brain. Now here I find something how the differences of rat mitochondrial proteomes. Now the proteomes is all proteins in the mitochondria. How we can compare these proteins? So we can use gel electrophoresis. So gel electrophoresis can be used to compare the proteins as in DNA electrophoresis and DNA profiling. We take the proteins, and these proteins we can load onto this gel and apply electrical field, and then positive and negative side. Then the protein molecules can move based on size and its charges. If there's a small molecules, they can move faster, and the large molecule they can move slower. On top of that, they have different charges. Some R groups are positive charge, negative charge. So that also a little bit add charge and the molecular size to run and separate the proteins. After separation, we can compare the band positions, and further, if we want to identify different proteins, we can check the bioinformatics, because it's a large libraries available almost all type of proteins. So if we know the size of the proteins and the species and the mitochondria, which organelles, we can check what are the proteins in the bioinformatic database. So gel electrophoresis and bioinformatics can be used to identify differences in rat mitochondrial protein. In that context, there may be a question that can be asked in bioinformatics and gel electrophoresis, right? So a little bit prepare for gel electrophoresis. Anyway, it is important to learn the gel electrophoresis, and also keep in mind bioinformatics is also very useful to identify DNA and proteins and other molecules, because it maintains large amount of database, right? Moreover, each organ possesses different protein composition, especially in the expression of proteins involved in the oxidative phosphorylation system. Now we check the brain, liver, kidney, and muscles like different organs. Their protein composition is different. Of course, that is different story, because if we take muscles, actin and myosin are proteins. So if we take actin and myosin, they are very high in muscles, and they may be absent in neurons. That context, protein composition different. But here they highlight in the expression of proteins involved in the oxidative phosphorylation system. Now, how we can handle this? Because if mitochondria is the universally, it's a one kind of organelles, and this mitochondria presence in the different organs, they are the same mitochondria. And these mitochondria use a same type of process like electron transport chain and oxidative phosphorylation, the expression of proteins involved in the oxidative phosphorylation system. So how we can explain their level is different. So we have to also look a little bit into that, because if we take the nerve cell, this mitochondria, and if we take cells in the liver, this mitochondria, there seems to be the same mitochondria, because the process and the product is at the end of the ATP production is the aim of the mitochondria. Expression of proteins involved in the oxidative phosphorylation system. So what are the proteins involved in the oxidative phosphorylation system? Enzymes and the transporters. Example, ATP synthase act as a transporter as well as enzyme. So if their expression different, only thing we can consider, it depends on the energy level. If energy level is high, the expression is the highest. If energy level is low, their expression is also low. So we can only argue in terms of expression, because energy level high, more proteins, energy level is less protein. It is important to note that neurons are post-mitotic cells with a lifespan similar to that of the whole organism. The post-mitotic is they never divide again. So it's end of mitosis. And the cell lifespan is similar to the organism lifespan. So they can't refresh, they cannot renew new cells. They have to keep the same cells until the end of the lifespan. Unlike in other organs, such as the skin or the liver, damaged neurons are not replaced during life. I mean, skin is one of the best place. So every like 3, 4, 5, 6 days, it's renewing the cells. The new cells are made, and in the digestive system, the epithelial cell again renew every 3, 4 days. And liver maybe rarely, not so often, but neurons are almost no. Stressing the importance of protecting systems, including antioxidant defenses, to maintain neuronal integrity and survival. Now you might be asked a question, how antioxidant uh, make this defense. So basically how antioxidant protect the cells and the molecules. Okay, I think again you can write the same answer. Antioxidants such as vitamin C can donate electrons. An antioxidant does not change into free radicals as it lost its electron, and this electron is taken up by free radicals, and the free radical is neutralized. So it can reduces the free radical by donating electrons. Post-mitotic cells such as neurons seems to be more sensitive to the accumulation of oxidative damage. Post-mitotic cells more sensitive, right? So give me 1 second. Okay. Post-mitotic cells are more susceptible to the accumulation of oxygen damage compared to dividing cells and are more prone to accumulating defective mitochondria during aging. So we can understand this. One function of mitosis is tissue repair. So tissue repair is important function of mitochondria. Mitosis is very important for tissue repair. If there's no mitosis, then there's no tissue repair. Here, same thing. Neurons are not dividing, so there is a damages are high. So damages are accumulating over the time. In addition, to bring a higher degree of complexity, neurons are exceedingly compartmentalized. I think it is again given here. What is compartmentalization? Compartmentalization is cell is divided into different divisions such as cell body, axon, dendrites, and even the synaptic terminals, even more specific compartment that are synapses. So there are four divisions we can find, compartment we can find in neurons. Consequently, a proper mitochondrial distribution is paramount to sustaining the energy requirement at specific locations within the different neuronal compartment. Now, why this compartmentalization is important? Because every compartment has a specific role. Cell body is the main part, control all the activities. Axon is the conducting system of the action potential. Dendrites is the receiving place for the action potential. And the axons are transmission of the action potentials. So dendrite take up the action potential, cell body control, axon transport, and the synapse transmit to the next neuron. Consequently, a proper mitochondrial distribution is paramount to sustaining the energy requirement at specific location. Now, the energy demand is also different. So, based on the energy demand, mitochondrial localization is also important. For example, synapse energy requirement is high. So, they need more mitochondria for exocytosis, endocytosis, opening of different channels like this. So we should keep in mind what is the role of this compartmentalization and why it is efficient for the cells and distribution of mitochondria. It is not so surprising that synaptic mitochondria, which need to sustain the energy required for synaptic activity, present functional differences when compared to non-synaptic mitochondria. Indeed, peroxide production was found higher in synaptic mitochondria of rats compared to non-synaptic cells. Okay. Now, why peroxide production is important here? Because peroxide is again related to this reactive oxygen species and free radicals. So peroxide is important again in terms of free radicals. That means energy demand is high, free radical formation is high. As a result, peroxide production is also high. Now I use this diagram again to give a little bit idea about this oxidative stress. Now you can see the oxygen molecule at this place produce the superoxide anion, and the superoxide anion is written like this, and this can further convert into hydrogen peroxide. That means peroxide production is directly proportionate to the anion free radicals, oxygen anion. After that, the hydrogen peroxide can convert into a OH, hydroxyl free radicals. The hydrogen hydroxyl free radicals and hydrogen peroxide and oxygen anion, right? This, this, you can see all these three can damage the molecules protein, lipid, and DNA. So this is again another system mitochondria is used to neutralize. If they form, there is another system is operating to neutralize those things. Right? So here you can see uh, this convert into hydrogen peroxide. Hydrogen peroxide convert into a hydroxyl anion. Hydroxyl anion neutralized by this glutathione system. In this research article, they don't mention about the protective system of mitochondria, which used to neutralize the free radicals. So this system is also available in mitochondria to neutralize the free radicals, glutathione system. But it's not mentioned in the article. But we keep in mind, mitochondria also has some kind of a mechanism to protect these free radicals. Here only they mention about antioxidant defense. Right? Unlike in other organs such as skin or liver, damaged neurons are not replacing during life, stressing the importance of protecting systems, including antioxidant defense, to maintain neural integrity and survival. So keep in mind, they have a good protective system, that is antioxidant defense. So we will be not asked about these specific defense systems like a glutathione system, but you should write if the defense system is asked, it is a antioxidant mechanism. So antioxidant are any kind of a system, they can donate electrons to neutralize the free radicals, right? So here it is given another mechanism like a ferrous ions, two positive charges. They can also take this kind of a catalase enzyme, then superoxide dismutase enzyme, like enzymes are there. But we will not ask, we don't want to say anything about this. If we ask, we will write only antioxidant. Donate electrons, neutralize free radicals. Interestingly, aging seems to be accentuate the differences between the two populations of mitochondria in 14 months old rats Respiration was significantly decreased only in synaptic mitochondria when compared to 3 months old rats. Okay, you should also keep in mind why they use animal for this experiment, because it's very difficult to do this kind of experiment in human, because analysis is difficult and it's unethical, very unethical use of humans. So ethical issues, use of rats are ideal. They are mammals and they have also a similar structure like humans. We can compare the result. On the other hand, animal experiment is also unethical certain level. This also keep in mind in case something asked about the animal experiment. So they use old and young. So old and young, 14 month and 3 month. So 3 months is a young age and 14 months is old age. So assuming that with the old rats, they also have some more free radical formation and neuronal damage. And they found respiration was significantly decreased only in synaptic mitochondria when compared with 3 months old rats. I think this also give an idea. Synaptic mitochondria are more susceptible for oxidative damage. Why synaptic mitochondria? Because synaptic energy production is high. Energy use at the synapse is high. So even compartmentalization, dendrites, cell body, axon, and synapse. Dendrite, cell body, axon, synapse. So this confirm the highest energy production we can find in the synaptic mitochondria, because in this experiment their respiration was significantly decreased. That means energy production decreased, more damaged. Besides, a higher susceptibility to calcium insult was observed only in synaptic mitochondria of old animal. Okay, what does it mean? Higher susceptibility to calcium insult observed only in synaptic mitochondria. Now, calcium insult is, we know that voltage gated calcium channel in synaptic presynaptic membrane is the most important transporter, because calcium moves in, then you start neurotransmission. In this case, the calcium influx is impaired. When the calcium influx is impaired, calcium cannot bind with the synaptic vesicles and it cannot move synaptic vesicles. So if you ask what does it mean by higher susceptibility to calcium insult, the synaptic presynaptic calcium voltage gated calcium channel opening and moving calcium inside is impaired. As a result, neurotransmission is decreased. Right? So if they have 1,000 calcium channels, maybe some not working, so less calcium channel is working. In non-synaptic mitochondria, oxygen consumption was not significantly affected by aging. By both population of mitochondria generation, higher level of peroxide in 14 months old animal compared to young animal. Okay, now non-synaptic neurons, they are not much, energy demand is not much like synaptic. So in this sentence, they say it's not much affected because they are not much energy used. Mitochondria possesses a residual genome approximately 16 kilobase coding for 13 proteins essential for mitochondrial respiratory chain function, which make them unique organelles carrying autonomous DNA. It appears that the quality control of mitochondrial replication is not as efficient as nuclear DNA, resulting in an increased risk of mutations. So mitochondrial DNA are more susceptible for mutations. One reason is they are autonomous DNA. That means they have no controls. They are controlled by themselves. So their replication and other things is less controlled by the factors. I think cell cycle. You should also look a little bit about cell cycle, AS level. In cell cycle, DNA replication happen at the S place. After S space, there are control system. It can proceed to the next level, and you can check the nodes. The cyclin and cyclin dependent kinase, cyclin and cyclin dependent kinase are the proteins which regulate a cell cycle. In that case, cell cycle is mostly the DNA replication. If DNA replication is perfect, cell cycle take place. So control system of cell cycle mean checking the DNA quality. So mitochondrial DNA, they don't have such system like in other nuclear DNA. So they are more susceptible for mutations. Little bit read notes about the cell cycle for this part. Fortunately, to avoid the accumulation of such mutations, mitochondria are remarkably dynamic organelles that involve and fuse in order to maintain homogeneous mitochondria population by content mixing mitochondrial DNA and metabolic proteins, quality control and distribution of mitochondria within the cells. Now, one mechanism is there. Mitochondria can replicate. Cell can't replicate, but mitochondria can replicate, right? So the neuron can't replicate, but mitochondria can replicate. So when mitochondria replicate, they can fuse with the other mitochondria. So this fusion will bring some kind of a better quality of mitochondria. If there's a bad mitochondria, when it fuses with the good mitochondria with less damaged one, so it can again function properly. So this fusion system is again protective system. They maintain quite good quality of mitochondria. It's called a fusion fission. Activity is also integrated with the mitochondrial quality control pathways, allowing the detection and removal of aged or damaged mitochondria through the specific form of autophagy and mitophagy. So it's a kind of programmed cell death. So mitochondria can induce death if there is a damage. It is not well understood according to this article, right? So basically, lysosomes are involved, lysosome fused, and then they can destroy the organelles. Okay, finally, we have finished the articles. The summary is when mitochondria fuse, they mix their membranes, matrix, and their membrane space, including all their content and mitochondrial DNA. After this mixing, mitochondria can divide, sharing quality equally. There are new content between two daughter organelles.
Basically, the mitochondria binds and the good organelles are the ones that remain in the new mitochondria, right?
Yes, it says that if this one is badly affected and this one is a quite okay one, when they fuse again, we can understand uh, the damage. We can't prevent the damage, but the new one, which do not have much damage, they can still maintain a good quality again. So then this fusion method can still it's a kind of a uh, removal of the bad mitochondria. I think, for example, one boy is not studying well in the class. If we move with the boys, they are studying well. Eventually, he's also learning much better because he has to do the same thing like that. He can remove these bad qualities and increase the good qualities. When a damaged mitochondria is detected, it is eliminated from the fusion-fission cycle by mitophagy, guaranteeing a homologous and healthy mitochondrial population. That means there is a system they can remove the damaged one and the new one stay. Thus, it is not surprising that defects in mitochondria dynamic and mitochondria quality control system may lead to cellular impairment, which was proposed to be involved in the process of aging and neurodegeneration. Conclusion is mitochondria are the most important place for rose generation and damage to the cells. With the mitochondria damage, the energy production decrease, then cells are not functioning. So if there is a defective mitochondria, this lead malfunction of neurons, especially the transmission like things, which can cause diseases. And mitochondria is specific for uh, this kind of a damage because oxidative phosphorylation generates large amount of reactive oxygen species. So compartmentalization is again important to maintain a different amount of mitochondria. As a result, only certain places are affected. In the rat experiment, the synaptic mitochondria are more affected because they have more damage. Okay, this is the end of the article. I think we guessed many areas, potential areas for question. I will send again these slides. You can check. You can read article again and also check the basic things like what I mentioned: the structure of mitochondria, respiratory chain, cell cycle, brain imaging, like these things again, gel electrophoresis. So revise again the notes. You need this information to write answers.